So happy to have you all there. We've got really a great representation from around the world here. I'm super excited to have you all joining us. Just for the record, I'm going to be recording today's session so that more families can learn and follow along with the update. I'm mostly going to be having Michael on the screen, so you guys won't be showing, but we want to be able to ensure that lots of families have this access to this information. And because this is a really exciting meeting. We're at a really exciting point in the SCN8A community. For those of you who don't know me, I should always back up. I'm Gabi. I am mom to Elliot, who is 12 and sitting with me here today. He likes being on Zoom meetings and practicing his head control while we do that. He actually does not like that, but he does it. We're here in Washington, D.C. I am the executive director and co-founder of the International SCN8A Alliance, and Dr. Michael Hammer, and Jayetta Hecker are the co-founders along with me. We are all impacted by SCN8A. I'm a mom, Michael's a dad, Jayetta's a grandmother, and we've been working for over a decade to really advance work in SCN8A, and we really feel like we're at a pivotal moment in SCN8A. You know, in the last few years, we've accomplished some really incredible things in SCN8A, including the publication of a global consensus on SCN8A, which we worked with more than 40 doctors and 13 families from 16 different countries. So truly a global project and collaboration. And that's really been a game changer for a lot of families who have clinicians that don't really understand or know SCN8A. And, you know, we are going to be talking a little bit more about how we continue to advance that information and share with more people. In addition, after the consensus was published, you know, we was very clear that we're still a lot of unanswered questions about SCN8A. So we started working towards a research roadmap meeting, which we held last August, which was a fantastic gathering of more than 40 patient advocates, clinicians, researchers, and pharmaceutical companies coming together to chart a path for the science in SCN8A. And Michael wrote a summary of that meeting and the consensus that came from that meeting, which should be published in the next few weeks, hopefully. And now we are starting to do a very thorough strategic planning process, and you'll be hearing more about that in the coming weeks, because we will be asking community members who want to be involved to help us think about how we chart a path forward for the Alliance and supporting critical research and advancement in improved care and therapies for SCN8A. So that's my very brief update about what we're doing and where we're going. And today we're really going to hand it over to Michael to share a bit of a research update and a research opportunity that the SCN8A community is really lucky to have, and is going to touch on one of the biggest gaps that were identified from the research roadmap that Michael will tell you more about. So I think the first portion of the meeting is really going to be focused on Michael, you sharing and talking, and then we'll jump into questions and hearing from people. I think people can chime in if they have questions along the way. Feel free to use the chat or raise your hand or anything like that. But I'll hand it over to you, Michael, to share a little bit more about your thoughts on how we use the time today.
Yeah, no, absolutely. I think that's perfect. I'd like to give about a 40-minute presentation. Elyse and I will tag team on this because Elyse is our clinical coordinator for the registry and for the project I'm going to describe. I'd love to get your feedback on it and hopefully get your participation, because you'll see as I discuss all the progress we've made and the gaps that still remain in getting better treatments for our kids. This is an important way to fill one of these gaps. So I think, let me ask Gabi if I could share my screen.
I was just working on that. I think you could try again now.
Okay, yes.
Okay.
All right, so the topic today is constructing a longitudinal profiles with caregiver and clinician reported data to accelerate improved treatments for SCN8A epilepsy. This slide is a cover slide, but it kind of covers the whole meeting. So if you fall asleep in 5 minutes, you'll have heard most of it just on this slide. Basically, what I'm showing here is a longitudinal profile for a single patient. And what we want to do is incorporate lots of data from each patient, especially daily seizure tracking and medication data, to be integrated with clinical data and electronic medical records, developmental milestones, quality of life assessments, and integrate all that data into a single pipeline so that we can make associations between how you're doing with respect to seizures and what else is going on in your life, and then looking for statistical correlations to identify what is making you better, what is making you worse, and what the best advice we could, the best information we can convey to your doctor. So it's basically constructing a longitudinal pipeline, comprehensive understanding of individual patient experiences and outcomes, and getting to a more personalized treatment plan and better outcomes. And Gabi already described a lot of the things I'm going to cover, but let me just give you an overview of the talk. So we're going to talk quickly about what Gabi already mentioned, the 12 years of rapid progress, but the critical gaps that still remain in understanding of the disease and how to treat it. The roadmap that Gabi referred to, which was a research mode map in Boston, where we identified the most critical gaps in SCN8A knowledge and identified research priorities to fill those gaps. I'm going to focus on treatments, and working group 3, which you'll hear about, was all about improving current therapeutics. And I'll explain what that means in a moment. And then I want to cover some things that a lot of you have participated in called citizen health, which you signed over permission to let your medical records be used for research at the same time you were getting access to all of your electronic medical records for your use in your home and for you sharing with your doctor. Despite all the electronic medical records and all the things that we know from the doctor's office, there's still a major hurdle, and that is we're not documenting daily seizures under any of the methods we're using. So we've designed a prospective research project to cover that gap. We're going to be using an app called Helpilepsy that you can download for free, and we want to describe how you can make a difference to push and accelerate progress in our field. So the rapid progress is just kind of shown here in a little infographic, starting back in 2012 when we identified the gene that causes SCN8A epilepsy, the registry that was launched, the mouse models, the iPSCs, the incredible amount of great attraction to the field we got from many scientists and doctors who made case reports and cohort publications. And you could see down in the lower right, since 2012, you can see the enormous number of publications that have come out in the scientific literature, over 470 publications in 12 years, motivated by the discovery of the gene and by the disease that we're all suffering from. And then we, in more recent times, we have laboratories developing genetic therapies. We have clinical trials from 2 pharmaceutical companies. And then, as Gabi mentioned, a very important step, the diagnosis and treatment guidelines consensus. Now, that was based on our current knowledge, the knowledge at the time, which probably would be up to 2022, 2023, all the medications that were being used to treat and what doctors could agree upon in a consensus sort of format. And so far, there's not a lot we can say consensus-wise other than oxcarb and carbamazepine were the favored drugs for gain of function. And that's not the fault of anybody. It's just that we're still early on, and most doctors haven't seen enough patients to know what's the best treatment protocol. So what we want to do is accelerate that very next step. So let me just say that despite that great progress, we still don't understand the disease mechanism fully. and I've bolded a couple of things I want to talk about today. The anti-seizure medications, I'll refer to them as ASMs, are inadequate and they're not selective. Polypharmacy is not well studied. It's hit or miss. Polypharmacy is the use of more than one drug at a time. Treatments for non-seizure phenotypes and comorbidities are lacking. Lack of strategies to deal with high premature death rate. Lack of biomarkers to track disease progression and treatment response. And there's very low rates of seizure freedom in our cohort, in our population. Only about 15% of patients claim to be seizure-free for 6 months or longer. That's a very low rate, and we need to do better. So we organized a roadmap meeting to attack these gaps, to identify them, to get consensus on the gaps and on the research priorities going forward. So the research roadmap took place last August in Boston, Massachusetts, and it involved a wonderful group of clinicians, families, researchers, and pharmaceutical representatives. And we had a very productive discussion for a couple of days where we identified the gaps in our knowledge with the goal of determining the future research priorities that both clinical and laboratory scientists would pursue to advance the field. And as I said, the participants were from all the stakeholders in our area. And what I'll do now is I'm just going to share a couple of the caregivers' concerns and how they were accommodated by the clinicians and researchers. I'm just going to give a couple examples because the meeting really did center on the caregiver concerns and how clinicians and laboratory scientists were responding or aligning with those concerns. So this is just from a very extensive survey that we put out to caregivers where we had over 175 responses from families like you around the world. And then something like 46 clinicians responded and 23 scientists. So it was a very, very successful survey among the stakeholders. And there was also biotech companies that were interviewed, and we were able to compare all the stakeholders' concerns, what they thought the gaps were, and where we think we should all head. So, for example, when we asked caregivers what treatments warrant further investigation, there was a lot of excitement about alternative therapies and combinations of anti-seizure medications and meds for disruptive behavior. When we asked clinicians the importance of research to improve treatment, clinicians felt that first-line and adjunctive ASMs were the most important. Adjunctive just means, again, drugs that would use in combination with first-line drugs, and also to optimize polypharmacy, again, the use of multiple drugs. When we looked at what caregivers cared about when it came to controlling seizures, they wanted more effective anti-seizure medications and combinations. Again, I'm hitting on the same themes because that's the theme of today's talk. And laboratory scientists were more interested in selective NaV 1.6 therapies. In other words, making the channel function more properly with therapies, mainly genetic therapies, but also new small molecules and repurposed drugs. So we all had similar concerns. There were some differences. Obviously, we're different groups of people doing different things. So it was great to see the alignment among the stakeholders. Now, when it came to caregivers, what do they care about with research priorities? Which of the following is a bigger research priority for you? And improving seizure control, about 60%, improving non-seizure outcomes, about 40%. So we did have a big section of the meeting talking about non-seizure outcomes, which I won't address today. That's something for a different meeting, but it was not ignored. It was something that everyone realized was extremely important. And in terms of controlling the seizures, caregivers ranked getting better effective anti-seizure medications as the top priority. And again, combinations and off-label medications and drug repurposing were all very important. But you could see the ranking in this graph here. And then finally, assume you're in charge of distributing limited research funding. Which areas of research is your top priority? Interestingly, 74% of people said gene therapies, despite the longer term goals. It may take years to get those gene therapies, but still people were interested in new drugs. in the short term, the 24% who answered that, and then other 2%. I'm going to talk about the short term and the long term trade-off today. So, yes, we want better smart drugs and gene therapies, but we know that's going to take years before we get access to those new treatments. So, what can we do immediately to improve care? That's what our next discussion will be about here. So, given all the concerns, the surveys, the results, the discussions at the roadmap meeting, we broke up into 5 working groups that represent, we thought, major gaps in research areas. So again, the working group 1 was advancing gene therapies, development and delivery. Working group 2 is non-seizure outcomes, mechanisms, management, and therapies. Working group 3 was improving current therapeutics, and that's what I'm going to focus on today. First line, adjunctive, and combination and alternative approaches. The 4th working group was the application of biomarkers, which are molecules or measurements that you can take that track the disease progression and the treatment response. That can be measurements taken from wearable devices, EEG, transcranial magnetic stimulation, blood tests, things like that. And working group 5 was looking at a more holistic view of the disease, the whole brain, the whole body, what brain regions, cell types are, and circuitry are involved in the disease, and how and neuromodulation could detect what's going on in the brain, and then the epileptic body, what kinds of things are going on in the entire body that are due to SCN8A. So this is just a quick look at all those 5 different working groups, what their goals and their gaps were. I won't go into all of them, but I do want to point out, number 1, that the smart drugs, the things that are going to come to us in the future, the gap identified by that group was current drugs are limited in their ability to control seizures without significant side effects and are ineffective in mitigating a wide range of comorbidities commonly experienced by patients. The goal is to develop transformative therapeutics that are effective in reducing seizure burden and with fewer adverse side effects. And I have a little mouse model over here because I want to point out that most of this work is preclinical. That means it's being done in animal models before it's being done in humans, although there are some exciting advances in SCN1A and other related fields where they are actually trying these kinds of things in the human at this point. So there's hope, but it's going to take time. The small molecules that you're hearing about in the Neurocrine and the Praxis trials, although they're ongoing in humans, there's still a long way to go to get FDA approval and get the drugs to market. So the working group 3's purpose, like I said, was despite the availability of all these anti-seizure medications, there are high rates of refractory seizures and little known about optimal treatment regimens. And the goal is to improve therapeutics on a shorter time scale than realistic for novel therapeutic strategies, i.e. gene therapies. So what was I'll give you a little bit more about the working group 3, because this is what directly led to the prospective study I'm going to talk about today and ask for your participation. So the background is that clinicians have been challenged to prescribe first-line medications or change their doses by a trial and error process. As you know, when you go to the doctor will ask you how you're doing. Are you getting better? Are things worse? And let's see what we need to do to either change a drug or change a dose. And there's no handbook to go to. It's trial and error, and it's based on the doctor's clinical experience and what they know about what's been published and what's been shared by the Alliance and the consensus process. So that's all very positive, but it's still very much a trial and error process. More than 75% of patients have failed 2 or more drugs. So that's known as medically refractory if you fail 2 or more drugs. And so 75% of us in the registry data have failed 2 or more drugs. That's a huge failure rate. And 85% of patients are taking multiple drugs, polypharmacy, which complicates our efforts to determine the best first-line therapies. If you think about it, if you're on more than one drug and things get better, you're not always sure which drug is doing the job, or if things get worse for that matter. So you have to guess as to what to wean, what to add, and stuff like that. So we want to do better. The process of choosing the first-line medication is made difficult by virtue of the fact that very little evidence-based data exists to support selection among the multitude of drugs on the market. And the multiple potential sources of data can inform clinical management, include things like the registry and electronic medical records. I'm going to talk about both of those today. And these sources collect seizure frequency data, but only a few times a year or when you see your doctor. So one of the things I'm going to point out is the baseline for knowing how you're doing is your seizure frequency and if it's going up or down. Doctors generally use a rule of thumb that if a drug is working, it means your seizures have been reduced by 50% or more. But we don't have a good count of your seizure frequency. So that's what our project is going to be designed to tackle. And the question I want to ask before we go into the prospective study is, do the registry and the electronic medical records that we have, the state of the art in terms of data collection, do they provide the necessary fine scale data to determine the efficacy of particular medications or medication combinations? So I'm going to tell you what we've been doing with citizen data to try and extract as much information as we can from records that the doctor is keeping in their office, and oftentimes will share with you, but certainly has been shared with the Citizen Health Project. Now, I don't know for some of the people who have come along more recently, if you've heard about this project, but both TCSF and our side, our alliance, have been asking for people to participate in this and share their data with us, the Hammer Research Foundation, and the Alliance, so that my lab can get access to the data and do analysis. And that's exactly what we've been doing for the last 2 years. So the mission for the Citizen Health is to use the power and shared experiences of families to provide relief to patients and families navigating rare and complex conditions. You centralize your medical records from every clinician and hospital in one place. That's what the Citizen Health will do. It centralizes your medical records, and then it shares your health data securely with researchers working on treatments for your disease. The researchers access comprehensive and longitudinal real-world data for unstructured medical records, for natural history, burden of disease, endpoint, the selection, external control arms, and long-term follow-up. And then what we want to do with the data is create longitudinal studies that show burden of disease, progression of patient symptoms, and genotype-phenotype correlations. For our disease, over 70 families have joined and agreed to share the data with the Hammer Research Foundation. I'm going to tell you about some of the things we're learning from the citizen data. And in the end, I want to point out that it's still not sufficient for the purposes at hand with what the roadmap had pointed to in Working Group 3. So this is a couple of slides are going to show summaries of the citizen data. This is the average for the whole group of 70 patients. So it's not one patient, but it's the average for all 70. And it just shows you, the nice thing about the citizen data is we can start to look at timelines. So the age of a patient in years is shown on the x-axis, and then the proportion of different seizure types on the y-axis. And you can see at very young ages, focal seizures dominate and slowly are taken over by tonic-clonic seizures. And you can see infantile spasms and spasm-like seizures are also frequent early on, but they end by the age of 5. Meanwhile, absence seizures increase over time. So we get this kind of view of the progression of the disease on average. The citizen health data are also very nice in that we can look at what we already knew. The disease has so many comorbidities, and we can look in the doctor's notes to see what those comorbidities are. They match very nicely what we see in the registry. So these are a host of different problems for respiratory, neurological, musculoskeletal, GI, sensory, immunological, behavioral, cardiovascular. That's why we have the whole brain, whole body working group working on questions like this. And then what's interesting also is we can see how many times you go to the hospital and why you go to the hospital for these unplanned visits or these emergency visits. And you can see over here on the left, most of the time it's due to seizures and status epilepticus. These are the most critical things that will drive you to the hospital, but there are a host of other reasons why SCN8A patients go to the hospital, including cardiac, gastrointestinal infections, other pulmonary and respiratory issues, and pneumonia. And you can see that they're all stacked up here in their relative prevalence as to why you would go to the hospital. Now I want to pivot and show you what we're doing to create individual profiles. And I'm going to spend a couple of minutes on this slide. This is the result of my lab taking 2 years of writing code to take the citizen health data, which is just medical notes. It's in text language for the most part, and we have to convert all the text language in the medical note, covering all of the different things the doctors know about medications, seizure history, hospital visits, adverse events, all of that, and convert it into zeros and ones. In other words, something the computer can understand. And from that computational pipeline, then we do in-depth analysis to try and construct a longitudinal profile for each individual. And I'm going to point you to the bottom panel, C, for a moment. What you have here is an individual. On the very bottom, on the x-axis, all those triangles, those are all the office visits over a period of 90 months. So that's several years. And you can see that there are a lot of office visits. And each of the office visits, information is collected by the doctor and entered into medical notes. And on the top, you can see the medications that were added in the order in which they were added. So levetiracetam, oxcarbazepine, gabapentin, phenytoin, valproate, and rufinamide were added in this order over this time period for this single patient. This is actual data. The patient here had an 1872W mutation, so we keep track of that as well. Then we have on this line here adverse events, and these are serious adverse events, and you can see where they correlate with the medications you were taking and with your seizure history, which is shown below. So the red dots represent seizure events, and the size of the red dot represents the intensity of that seizure event. And you can see we have tonic seizures, focal seizures, tonic-clonic seizures, all for this given patient. And then we have status epilepticus events shown here as well. And then at the very bottom here is developmental milestones. Green means you've gained a developmental milestone. Red means you've lost a milestone. And over here you can see we've got them categorized as fine motor, gross motor, and language. And finally, I want to point out that one of the things we've done is to say, well, what's working in terms of medications? And our first pass was to identify the biggest gap in seizures. So in this particular patient, there's a gap of several months where seizures were subsided entirely, according to the medical notes. You see there were still office visits here, but there were no reported seizures. So, you know, when you go to the doctor, they'll say, how have you been doing since we last saw you in the office? And you'll give an update on, well, my child hasn't had any seizures since I last saw you. And so that goes into the medical notes. Or my child had roughly 5 seizures a week, you know, for the last 3 months. So we get an estimate of the seizure count, but it's not an accurate estimate. Now, what we've done from all the data down here is that we can show the seizure index. So we ranked each seizure type on a scale of 1 to 5. And then for each individual seizure type, we can see how you were doing over the same period of 100 months or so. You can see here, focal seizures got worse for a period. Here we had tonic seizures, and you have tonic-clonic seizures going up and down. Over here is a heat map showing you our estimate of whether drugs were working for each of the seizure types. So here's those 3 seizure types. Here's 2 different estimates of whether the drug was working. If it's blue, it shows seizures are reduced by a particular drug. If it's red, seizures were exacerbated by a particular drug. And you'd see levetiracetam, as we know, has got a lot of red. That means levetiracetam is not actually working very well. Whereas valproate and rufinamide, in this particular case, worked okay for tonic-clonic and tonic seizures, not so well for focal seizures. So different seizure types respond differently to different medications. And we've been able to see all of this in the citizen health data. It's very, very helpful, but I'm going to show you it's still not enough. So I'm going to take you through a few more examples of patients who have these longitudinal profiles. We are happy to provide these to you if you participated in the study. And just note that the code that we've written to produce these longitudinal profiles will be similar to what we're going to do in the prospective study that I'm going to describe, so that we can provide you with similar kinds of longitudinal profiles of your seizure history, medication history going forward in time on a much more fine scale level. So here's a couple of more examples of patients. You can see here's a patient at the top who had a very tough time with seizures for most of the 100 months that we're tracking here. very different seizure types, lots of different medications, but yet there was still times when seizures got a little better. But I will point out that this gap is somewhat artificial because there were fewer appointments, but perhaps there were fewer appointments because the patient was doing better in that time. We've noticed the clumpiness of appointments, these of office visits, tend to be clumped around times when seizures are bad. There may be other things that will drive you to go to the doctor and make appointments, and we can see that in the medical record. And we've done some statistical tests to show that this clumpiness of office visits correlates with increased seizure burden. So here, despite the fact that there's fewer office visits, there are enough office visits to infer that there is a subsidence in the seizure burden in this period. Then we can look to see what kind of medications were being taken in that period. Here's another patient who had a very tough time early in life, lots of office visits, and occasional breakthrough seizures of different sorts later in life. And this was one of the two gaps, and this is the longest gap. So in the analysis I'm going to talk to you about, we're going to analyze the longest gap in this patient and see what was going on to decide what medications were working in the gap. So the next slide, I'm going to show you medications on average that were working for everyone in the citizen health data set in the gap. So I'm going to focus right now on monotherapy in the gap. In other words, which patients had a subsidence in seizures to the point of none reported in medical records, and which single drug were they taking? And it turns out for monotherapy in the gap, all the very dark parts of these bar were monotherapy, and you can see they're all sodium channel blockers: oxcarbazepine, lacosamide, lamotrigine, phenytoin, and carbamazepine. For those that were taking two medications in the gap, it's the lighter shade, and you can see that some of the sodium channel blockers were also administered as second drugs, as well as GABAergic drugs like clobazam and clonazepam. As interestingly, Epidiolex was another drug that was often used as in dual therapy and polytherapy, which is shown in the darker shade of gray. So you have both sodium channel blockers, GABAergic drugs, Epidiolex and Topamax or topiramate being added on as drugs in the gap that may have helped stop seizures completely. Now, this is not individual data, right? This is on the whole cohort together, but it's still valuable information that may guide doctors' decisions in the future. Just to show you some of the things, we've divided patients into those that had different seizure burdens, and we just called them mild, moderate, and severe. So if you had a low seizure burden, and we had a artificial cutoff for mild, moderate, and severe, but you can see we divided the longitudinal profile into two halves: the first half of your profile, the second half of your profile, just arbitrarily dividing it in half. And you can see the good news is the number of office visits goes down for mild, moderate, and severe in the second half relative to the first. The median time between office visits goes up, so you're going to make appointments less often, especially for mild, but also for moderate and severe. And then the seizure events gets lower for mild, very low. It gets lower for moderate. It also gets lower for severe, and the seizure burden also gets lower in the second half of profiles for all classes of severity. So that's good news in the sense that on average, treatments in the second half of the profile seem to be working somewhat better than in the first. Now, is that because the medications are working better, or patients are resolving slightly as they get older? We don't know the answer to that question because we don't know the natural history of the disease well enough to know. But we certainly want to pay attention to the treatments that are used that seem to be reducing the seizure burden over time. And I just want to point out that the major problem gap that we face in advancing or accelerating the rate at which we're learning about how to better treat the disease with the current drugs that we have is the accuracy of our seizure frequency estimates. We document seizure frequency at office visits. The doctor asks you how you've been doing since we last saw you. You give them an estimate of how many seizures you had per week, per day, per month. That's just an estimate. It's not a seizure count. Our graphs show the seizure intensity based on what you said at that doctor's office visit. It's not a seizure count. The mean office visit frequency is on average 2 months, although sometimes it's as long as 9 months. So there's recall memory problems. You're not going to necessarily remember unless you're documenting the seizure counts at home accurately. Generally, the doctor doesn't take that information in. I know that I documented my daughter's seizures on an Excel sheet for 9 years. And I would often try to show it to my doctor, and the doctor was interested, but didn't take it into consideration in their medical notes. They took my sense of what was going on into the treatment protocol, but it was a qualitative assessment. Again, we don't do any better in the registry. And the reason is we estimate seizures annually. In your annual updates, we ask you how you've been doing with seizures since your last update. And it's an estimate. And what we found is that on average, or the median number of seizures per month in the registry and the annual updates is 8 per month. That's an estimate of the seizure count from your recall of what's been going on over the past year. The citizen data, we only have seizure index, so we have no estimate of the actual number of seizures per month. It's a fraction because it's based on office visits, and we can't interpolate what's going on between office visits. All right. Now getting back to the purpose of the roadmap and working group 3. I know we've covered a lot of territory. We're getting close to the end of the presentation, and Elyse will tell you about the app that we want you all to use to fill this gap. So as I've already said, we don't get enough high quality, high resolution longitudinal data to document how seizures are changing in parallel with changes in medication regimens or other life events. For example, did your child get sick? Did they have a fever? Did they have a status event? How does that affect your seizure count? We don't know because we can't correlate them accurately. We focused in this working group on proposing a unified infrastructure to allow for efficient collection of high quality multi-source data that would provide the framework for improved understanding of the efficacy of single medications, combinations, and alternative therapies. So the specific aims to come out of this working group for research: design a strategy to collect granular data on seizure frequency in a prospective manner to advance knowledge about optimal medical combinations. Use of a well-vetted commercial application for seizure tracking with back-end data processing. In other words, can we have you use the app and us in the lab obtain the data that you've entered? So we needed a company that's already devised that system, and we found one. And then focus our work on efforts to merge the data, the daily data, with other sources of data that we get from the electronic medical record and from the registry questionnaires, and, as I will describe, wearable devices. So that brings us to where we are today. And you know, we've been advertising this project. We really want you all to participate. We've decided to use a free app that you can download right now called Helpilepsy, and we have the QR codes that you can go to get the Apple or the Android versions. I'm going to let Elyse talk you through it, but this is basically what you will see when you enter data for a period of time. The bar graphs show you seizure events, generalized tonic, clonic, focal, other seizure events. You enter in, let's say you have 3 seizure types, A, B, and C. Each day you go in to say, did your child have an A, B, or C? And then you also enter in your medications you're taking. Each line is a different medication. And the app will also ask you about sleep and mood. All right, Elyse, I want to turn it over to you right now to tell us about your slides that you created. I apologize, the slide format changed because I couldn't put them in my slide deck and not have your format changed, but you will recognize them. since you made them and sent them to me yesterday.
Hello, families. Happy Saturday. I am the clinical coordinator for Dr. Hammer. So when you email us, you're often talking to me. I talk you through problems that are happening or answer some of your questions. Um, I also have epilepsy, not SCN8A related. It's due to a TBI. I suffer from temporal focal epilepsy, and I've tried many of the medications that your children have tried, and am now on monotherapy with Xcopri. So today I'm just going to talk to you a little bit about, do you want to go to my first slide, Hammer?
Yes.
Yeah, there we go. Okay, so we are starting the first phase of a prospective study to get the knowledge gap filled of what's actually happening to SCN8A patients and their seizure frequency, because there's a large gap, as Dr. Hammer said earlier in the discussion, between what we're seeing reported in office visits and with the registry once a year. There's critical information there that we would love to have to better treat seizures in this population. This project is a bridge between both clinicians, families, and researchers to help fill in that gap. So next slide. The app that we use is called Helpilepsy. It's a free app that's been available through the Apple and Android store for many years. It's run by a company called Neuroventus. They also make a wearable device that we'll talk about later in the slides. But it is FDA approved as well as IRB. So we have a research board that ensures compliance of HIPAA and protects information appropriately. And this app is approved by them and also other research boards that allows us to see your seizure frequency, your medication history, as well as your mood and sleep, that we're hoping to find some correlations between specific medications or medications missed or just general life stress on a granular level that we haven't been able to see before. Daily logging in this app is very simple. I use it myself to track my seizures and log my medication. And it allows us on the back end. So we purchased a license to get the data on a very discrete level we are currently creating code to highlight. Sorry, my allergies are terrible. To highlight where the gaps in treatment are. So we can go to the next slide. Okay. Again, SCN8A is a rare disease or disorder, as we know, which means not all neurologists know what seizure frequency looks like in this population. Even people who have, or clinicians who have seen SCN8A patients haven't seen enough of them to really understand the patterns that arise within the population. We know because we have a registry of over 400 patients, but there's a large gap, and we're hoping to both empower researchers and you, the caregivers, to have better information to share with your care team so that we can get appropriate treatment. Because the first line that is prescribed for ASMs are not typically what works best. And as Idan has said in the chat, clicking a few buttons is a lot easier than doing an Excel sheet or a form. The app is relatively user-friendly and will give us a lot of information that we're going to share back with you once we move into the prospective study. It's 100% voluntary. The app is completely free. Let's see, there's a question. Is there an app to track medication and behavior? The app tracks daily medication intake. We don't have like specific behavioral questions in that app. There is a mood. It's a 1 out of 10 scale. So that could essentially highlight some of the fluctuations in behavioral concerns. Um,
We've
okay.
also got the surveys. We'll talk about the surveys too.
Yeah, and
Quality of life.
yeah,
Yeah.
there's so the other part. Oh, there's lots of questions coming up, and you can review what we give you. So that is one of the goals of the study, is to give you and empower you with a subset of data that you can bring to your neurologist and discuss very clearly what you're seeing, because that's one of the issues when we go into an appointment, right? You go once a year, once every 6 months. I mean, you often go, or at least I was going very frequently when I was first diagnosed. And then once you're diagnosed for X amount of years, you only see them every 6 months. And you're kind of giving an approximate of your seizure frequency. This is giving hard data that you can share with your neurologist and say, on X date, my child had Y amount of seizures. Here are the things that I think might have been contributing to it: poor sleep, medication change, missed medication, illness. There's places in the app to put notes. We will also, once we move on to the second phase of the prospective study, be asking more in-depth questions. You'll be meeting with me on a regular basis to fill out a survey where we ask in-depth quality of life questions that you will be able to share with your neurologist. So we're both getting something out of it. We're able to fill in the knowledge gaps and hopefully educate the greater community on the current ASM treatments and the effects on SCN8A, as well as giving you a powerful data set to bring to your neurologist so you can get answers as opposed to an approximate, because we know life is busy. It's very arduous having one or multiple children to take care of, and collecting data on a granular level empowers you to go to your neurologist and say, this is exactly what I'm seeing. This is when I'm seeing it. We need to make some sort of change. Here's what I've learned from these papers. Like, I'm always available. I will empower you with whatever research we have available currently, and provide you with papers that you can print out and hand to your neurologist and say, this is what the current research says. Here's what I'm seeing in my child. Help. So we can go on to the next slide. Helpilepsy is very simple. This is what we ask of the current participants right now. There's no additional surveys until we move on to the next phase, but it's just coming into the app, log your medication taken, record any seizures, note special symptoms. This is also missing, that we want to do mood and sleep, but it's a quick little icon. It's a 1 out of 10 scale. You use a slider and you go, terrible sleep, frowny face; good sleep, happy face; terrible mood, frowny face; terrible good mood, happy face. And then you move on with your day. Next slide. Is that, I think you added
Yeah, I'm going to talk about the 2 phases now.
okay. So I'm going to hand it over to Hammer to talk about the next phases, but the app is already currently available through the Apple and Android store. There are QR codes. I can drop links, actually, while he talks about the next couple of slides. I'm going to drop links for downloading the app into the chat, so you can just click it. And the only thing we ask you to do is send us an email at info@scna.net
Yeah.
so that I can add you to our study. You'll get a notification in your app that Michael Hammer has requested to share data, and then that's it. And then we can see you on the back end. We'll intermittently check in with you via email. And then once we move to the prospective phase, you're going to get regular graphics that will hopefully help show you where patterns lie, and then we'll empower you to bring them to your neurologist.
Okay, thank you, Elyse. That was great. we're going to answer questions. We're almost done. So I just want to tell you what our prospective study design is. The nice thing about it is prospective, so it's statistically more powerful than retrospective studies in that we can make predictions, test hypotheses. And so we're going to learn a lot. So the first phase is what we're in now. We're trying to recruit at least 50 patients. We're going to, once we have 50 of you using this, we're going to do caregiver training with webinars and one-on-one training. And then we're going to watch you for a month or two. And for those of you that are using it on a daily basis, we don't know how many of you will, but we hope to get about 20 families that are using it regularly on a daily basis. And it's not limited. So as many of you who want, but this is what we hope to get. And so this is the daily seizure and medication data that we just described. We can also talk about some of the wearable devices that we have an opportunity to use in the prospective phase. So once we have this 3-month setup, getting you all on board, then and we are meantime writing the code to combine information from you, from your EMR, and we've talked to doctors about how to get access to your EMR. That'll be something I'll discuss in just one moment. We'll also start quarterly surveys via REDCap, asking you specific questions about developmental milestones that have been gained or lost, and quality of life surveys. So we will have specific questions for those in the prospective survey, the prospective study, to ask you how we can, what information we're going to correlate with your seizure burden and medication history. And then we're going to integrate all of that into a pipeline and do statistical analysis to look for correlations. The wearable devices that we can add during this phase include Night Watch, which is a small band that you can put around your ankle or your arm that measures seizures at night, and it measures heart rate variability. And we're in discussion with some different sources for possible ways of having periods of time where you wear a headband and we monitor EEG at night and your sleep, NREM and REM sleep at night. So we'll get really detailed information on sleep quality at night during the 9-month follow-up. So that's our plan. The big picture is we collect data, we fuse the data, we do data analysis, and we get actionable knowledge that we provide to the research and clinical communities. Finally, just here is the QR codes, and we're going to open it up now for questions.
So there's a bunch of questions in the chat. Maybe we can start there, Michael. I will start at the top. Let's see. One of the first questions, Don, I think we'll come back to your research questions at NIH because that's a really good question. But to focus on the app to start, so there's a bunch of other questions. Is there a way to send information about seizures in another way than an app? Can the form that gets filled out and then get sent instead?
Yes, that's a great question. Hold on, the email address is wrong. We just changed our email address, by the way. So there's the right email address. So we know several families are collecting information themselves, various mechanisms, and we are going to have a meeting with as many of these families who want to join us, and Elyse can remind us when that might be, to discuss the best ways to get your data to us. And then we'll take your spreadsheet data in whatever format you have, we'll convert it into our own computational pipeline, ourselves writing code to make it all coherent with the Helpilepsy data. So for those of you who prefer to use a spreadsheet or another mechanism, we'll have a discussion on how we can harmonize that with Helpilepsy data from the back end and make it all seamlessly one data set.
Yeah. And I think Edon was just also pointing out that, it's a few buttons, and so it might be worth trying for people who are hesitant to think about it. There might be some ways to improve on the current mechanisms we use, which I'm guilty too of trying to figure out how to integrate tech into our lives, but it's so I encourage everybody to try it and see how they feel about it.
You want to switch to gallery view so everyone can see each other instead of looking at my face?
I also wanted to say I'm not very good at keeping track of the seizures. And then when I have been using the app, I was able to count back to the beginning of March and see that he had 19 seizures. And I was able to tell my neurologist that, hey, he had 19 seizures. This was the kind of seizure he had. And I mean, it was helpful, actually. So,
Good. That's good.
and I'm not
Perfect.
tech savvy either. So if I can do it, probably anybody can do it.
And anybody can feel free to chime in too. And then Teresa, this is a great question. Can we review what we input into it when we meet with a neuro? So I think some of those graphics that you shared, Michael, what's the frequency of being able to access those that dashboard?
I'm going to let Elyse answer that. How often can you access your dashboard,
So unfortunately, the way that Neuroventus shares the data with us is a little complicated. I can see that dashboard whenever I log in, but we don't intend to share. They're a little complicated and not that easy to read. So once we move in the prospective phase, that's when we're going to start providing you with simpler graphics. We're currently working on code to take the data set that Neuroventus, that's the company that created Helpilepsy, shares with us and make that into easily digestible and shareable graphics for you. So not currently, but it will be in the next phase. We're thinking maybe once a month. We could do more. We're still in the initial planning phases, so we're open to feedback if you're interested in more frequent. We're just still working on the code to take what Neuroventus gives us and turn it into beautiful graphics that we can share with you.
And Tammy, maybe you talk about how you access your information to be able to report back to your doctor. What did you do? You just looked back and saw counted?
Yeah, you get into the app and you go into the diary button and you just scroll backwards and it tells you every day. And then and underneath your days, like I would note that, hey, he was cutting molars and he started having more focals and I gave him extra this medicine. So that would be written in as an event, but I was able to scroll back as far as I started it and count because the seizures are in red. So, I mean, you can just count the red. It says focal seizure, or it'll say tonic-clonic seizure. And then some of the things are in gray. So if I had an incident where it looked like he was dystonic posturing, that would show up in like a gray. So it was super easy just to scroll back and say, hey, I wouldn't have had the information to tell my neurologist that he had 19 seizures because I don't keep track anymore, but I had that when I had my meeting the other day. And then we decided that, this is way too many. We need to increase, and then we're going to possibly be starting the praxis trial because they did approve us. I guess I got within the 30 days. She messaged me back again this morning. So, and that, I'll go back in and I'll probably add that. I don't know. How would I add that in, Michael, if I don't know if I have the placebo or not?
Well, you could just put it in.
Okay.
Whatever it is, just say praxis drug.
Okay, doke.
Excuse me, Michael, I have a question. This is Teresa. How are you? Teresa.
Hi.
So in my case, there's times that I would have to give medication to calm him down. So there's certain behaviors that happen. So the neurologist has said, give him clonazepam to calm him down, and hopefully that would prevent a seizure. So there's been time I've had to like add a little bit, try a little bit or something like that. Would that be part of
Yeah, that will show up as a blip on your medication line. If it's only something that's occasional, we can still use the data to correlate with what's going on.
And you would have to enter what medicine you tried and what happened with it?
I note it as under an event. So you go in and then it'll give, I have a whole list of things that I've done, like gave extra Vimpat, gave extra clonazepam, and then I'll write how much, and then it will save it in there for you. So then that it'll be there later. So if I gave him 3 clonazepam extra, because my son does the same thing, I have to add Vimpat and clonazepam a lot. So then and then it tells you, ask you what time, and it asks you if it was all day. So if you don't put down what time or all day, then it'll just put the time in that you go in and do it. So you have to change the time sometimes if you do it later, but it'll save it.
The problem I have is that it doesn't make a difference. See, my son is nonverbal, so it's kind of hard to like figure out what is going on with him unless he has obvious problems. So they don't work to prevent seizures. Don't know if they even do anything at all because he has autism. So it's like, oh, okay, did it work? Did it not work? Then I ask the doctor, then they don't know. They're like, I don't know what to tell you. My son is older, so I've been through many medications, etc. So it's kind of hard for me to, I mean, I hope it would help everybody, but at the same time, my son's still having seizures. Like they're still, you know what I mean?
Yeah, well, I mean, even difficult things like that, it's worth trying because maybe we can learn something that you weren't seeing. Maybe there's a pattern there that we didn't know about.
Okay.
Yeah, that's a good point, Teresa. So there's a good question here, like, should we still be using the app if we're only seeing absent seizures and are not on any meds?
Are you doing anything to try and reduce seizure burden from absent seizures? Because I think anything you're trying to treat, any treatment you're trying, it's worth documenting. And if it's just absence seizures, sometimes people take meds for absence seizures, so that's fine. If you don't have meds, you might want to use the app. It might not help us with our research, but it might help you to correlate sleep, mood, and his absence seizure frequencies and things like that. And we'll still be able to see your data if you share it with us, and we can then have a better sense of what's happening with you when we have one-on-one conversations. So I think there's advantages to just knowing more on a granular basis and adding that into our discussion in our community.
And then if you do add meds in, you get to see the pre and
The pre and
post.
post, yes.
Yeah.
Yeah.
I had a message come in asking about, well, how do we know what the seizure types are and how we classify them? So I think that's probably a pretty good question to tackle here.
Yeah, we want to onboard you with send us a video. If you think there are multiple seizure types, you can put in the Helpilepsy app, and maybe Edon and Elyse, you can tell me the exact way you do it because I'm not using it. But how do you set it up so there are different seizure types, and do you have to label them initially, or can we go back and label them after we watch your video or get confirmation from your doctor?
When you put in the seizure, it asks the seizure type. So, I mean, typically we're hoping you get some sort of information from your physician of the types that you have. You can include a video in the app that can help go back historically, but you know, it is flexible in that you can go back and identify the seizure type. But you know, again, we're not clinicians. We're doing our best with the information that we have to identify the seizure types. It really is helpful to have classifications from your physician that you can use as you go through the app, if that makes any sense. So you can just put seizure event and then go back historically. Yeah, you can just like Edon said, we can help classify, but again, that's not like it really is up to your physician to classify via EEG and confirm. I mean, we have a great subset of knowledge and are pretty accurate, but we can't prescribe anything. So
Okay.
Can you show us an example of the app? Can you walk us through the app on here or no?
We, I don't, there's no way to like do it. I can open the app on my phone. There's no way to like access the app via PC. Um, let me open it. Let's see if this. Oh, I got a
We
lot.
also plan to have some training sessions. We'll have videos. The
Yeah.
company itself has a training session that we can share with you, but we will make sure you know how to use the app and walk you through it once you decide you want to try it.
I'm gonna log into the app right now. I just have to, it's asking me to reset my password, so give me a moment and I can kind of show you the screen. I don't know how helpful that will be, but we'll see.
On the seizure part too, I think there's also like a place where you can put other, and there's been times where I'll write, it appears that he's having dystonic posturing. So if sometimes I'm unsure too, like with a focal and some of the other things that he does, because they all kind of look alike. So I'll just note in there that it looked like he was dystonic posturing or possible focal seizure. It was, but right now my phone just clicked on and said, hey, how are you feeling today? So that would be the cue for me to press, Liam's doing good, with a smiley face. Um,
There basic information that we can email directly, like so like seizures, when they happen, one medication, and what else? To start that route.
I can't load information into the app for you. You would need to log in and then start
You could
log.
walk her through it.
Yeah.
Yeah, the idea is that Elyse will be available to answer on any questions about any time to try and get you to do it yourself in the app.
So you log into the app. I don't know if you can see this. I wish if I had an Apple, I'd be able to share, but I have an Android. And then you have your notification. So when you start the app, you put in your medications that you take, so that you can select that you've taken them. And then, I haven't been great about updating my app. And then there's this diary. So at the bottom, there's the home. That's the screen you just saw. So you have your medications and then you log whether or not they're taken. You'll have your graph of your seizures. And then you have this thing called the diary, which you fill out each day. And then you have your mood and sleep. So if we click on mood, it's just a 1 out of 5. You can slide this bar. And then your sleep is the same. 1 out of 10. And then any seizure events that you input, so you have your medications, whether or not they were taken. Everything's flipped because you're looking at it on a screen.
We
And
could see it right.
you have your current, so this is all the medications that you take. You can load them in here with this little plus. This is add a new medication. And then you can see, this says, this is a new treatment for epilepsy. If it's for something else, you can click, so like any of the mood ones, and then you can select, these are different, like, what is it for? Is it for headaches? Is it for anxiety? Is it for something else? And then this validate is just like an okay button. Confirm. Okay. And then in your settings, you can do your seizure types. You can do seizure videos. The trigger model is like, what do you think causes different seizures. So for me, stress, dehydration, and illness, I mean, these are very common seizure triggers for anyone with epilepsy. You can add like additional long-term conditions that contribute to it. So like chronic infection, short-term issues. So like stress, for me, overheating or too much sun exposure, combination of stressful events that might cause a seizure. So it'll ask you like, do you think there was a trigger? So for me, dehydration, stress, or fever. You can log any appointments that you have in the future with your physician. You can get, so once you have more data in here, it will also give you reports that you can download and share with your physician directly from the app. We're going to give you a different report that'll be more simplified than what the app provides. What else? You can also add, so if you are in the prospective study, you may be getting a device called Night Watch, which is a nighttime seizure monitoring device. It's an armband that measures heart rate variability and movement through a gyroscopic sensor. You can connect it directly with this app. And then you can have notifications set so that it will remind you to put in your notifications, that it will remind you to take your medications or to input your medications. And then you can also do an additional reminder, when do you want to be asked to put in mood and sleep? Let's see what else there is there. That's pretty much it. It's a pretty simple app. Are there any?
Yes, it does look very simple.
It's very simple. Sometimes the adding of medications can be a little complicated. Idan, if you want to speak about that, because you have a more complicated medication schedule for Michael than I have. I just have, two medications.
Sure. Let me see if this works. Just a sec. I need to change my settings here in order for Zoom to be able to share my screen. Do you see my screen?
Yes.
Yeah.
Okay, great. So this is the homepage for the Helpilepsy app. This is where I track my son's seizures and medications. Up here on the top where it says My Notifications, when it's medication time, once in the morning and once during the night, just like we saw in Elyse's screen, there's basically a button to click for every medication that the child needs to take. That's pretty easy. I get a notification on my phone, and then I click on the medication button so that the medication was given. Let's go to the treatments tab. So here in the treatments tab, we can see all the different treatments, all the different medications that Michael is receiving. It's very easy, to add a new medication or edit the intake and hours and so on. And there's also a history of all the different medications that were given and when they were changed. So that's regarding medications. What you need to do is basically enter which medications are given, and also gives you a reminder, which is neat. And then when your child has a seizure here, we can see the yearly seizures by month. If I click here, add seizure, when my son has a seizure, I go here. The time is currently the current time. Here you can select the type, so tonic, clonic, so on and so forth. Felt it coming, not relevant for my son. During sleep, that's also something that I select, whether or not the seizure was when he was sleeping or not. And the duration, I think duration is also interesting because that's also an interesting thing to track. There's a bunch of other questions, but they're all optional. And then I go ahead and click save. My son has a seizure. Yeah, confirm. So this is if you want to use the app very simply, just medications and seizures. There are other features that Elyse has shared, like mood and sleep and so on. But I have not been using those. And so this is really a basic way of using the app, and it's really only a few minutes every day. It's not a lot of work at all. It's very convenient, and yeah, this is what I use. And here in the diary page, you can see the different medications that were given, to see if you, to see the history of meds and seizures. So you can see what happened on a given day. Like, this was not a great day, not a great day for Michael, but yeah, this is all saved, so you can scroll back and you can look through it. And if you have an appointment with your doctor, you can go ahead and show this, and it's very easy.
So I have a question. This is only going to be as valuable as the amount of time you are observing your child, right? So if you only observe your child 1 hour a day, then we're only going to get that small window. How do you guys think we should? I think we're going to have to ask people on average, how many hours are you monitoring your child on the weekdays or the weekends if things change for you due to the work week, so that we can actually get a seizure frequency based on the number of hours that we normalize by the number of hours you're watching your child. Is that make sense? Or you think there are seizures you're missing, Idan, for example?
For me, it will be very easy because my son has 3 to 4 in a month, and they all happen the same thing, and no medicine has changed forever. So it's just easier for me to just give you an update. Okay, from January, February to May, same 3 seizures, 4 to 5 minutes, medications. So that would be a whole lot easier for me to keep another thing to keep track of. That's why I'm not crazy about an app.
Yeah. It's not going to be perfect for everyone, but for those that have regular seizures and multiple medications, probably very useful.
I have caregivers that help me. And we have an erase board. So like, if he has an issue or something, it gets written on the day and what time. So even if he has, goes to the bathroom, it gets written down. So if he has a focal seizure, they'll write down, hey, this happened at 3:00. It lasted about 45 seconds. So we have, and the girls are trained to know what the seizures look like, and stuff like that.
I think consistency, whether you're only monitoring 4 hours a day or 8 hours a day, the consistency would matter because we want to see changes in seizure frequency. So we just want to make sure that we're normalizing to the number of hours a day. But if you're consistent and it's usually the same number every day, or maybe it changes on weekends, as long as we know that, then, and I think we might have to ask that separately because I don't think the app is monitoring that. Is that correct, Idan?
Yeah, that's correct. I think we'll need to ask every family separately, what is the consistency that they use to track. I mean, sometimes the child is with a caretaker or not with a parent, and then it could be difficult to track the seizures. So it's going to be different, I guess, for every single family. And regarding whether to use or not to use the app, so the way I think about it is that I'm not a researcher, so I can't contribute through research. I'm not a philanthropist, right? So I can't, donate huge sums of money for SCN8A. But what I can do is I can donate my data, and that I do have, and that is my way of not only helping my child, but helping other children with SCN8A. So this is my way of giving back, and other families that are also looking to give back in some way, this is your way of sharing your child's data in order to help your child and help other children.
That's a beautiful sentiment, Idan, and I think very motivating. And it's so true. We need you as researchers. We need you guys, and hopefully we can make it worth your time as well. That something will come back to you. We see it as a win-win, two-way street. Mary, you made a comment. I didn't see what it was, but I always love to hear from you. Probably something about benefiting society or something.
Sort of. I'm trying to make lunch too, Michael, so I'm off. I'm just saying that even though I think as parents, most of us look at, oh my God, one more thing to do, but I'm begging people to use this because it's benefiting the entire group, even if it's a little additional concern to start something new for you, or you're not comfortable with electronics or tech or whatever. Please, please, please take advantage of this. It's not just helping you and your family, it's helping every single family in this group, because we're sharing this information with our researchers and they can look at it and look for patterns for medication. And gosh, we never noticed this before. You, just like Aiden just said, we are helping everybody. So please take the time. I get nervous with tech stuff. But for this app, I wish I could use it. My son has absence seizures, but he hasn't had any in a long time, and we're not using medication. So I feel kind of bad. How awful is that? Oh, I wish I could use that just to contribute something. So please, really take on something that is outside your comfort zone. Do it for the benefit of the whole group. That was my message, Michael.
Thank you. Beautiful.
Yeah, and I think something to keep in mind too is that I think Mary said it perfectly. Is we feel so helpless in so many ways, and like we can't do anything. And like Aidan was saying, this is how we can contribute to a better tomorrow too for kids that come. This is how we've learned. The registry started to set that stage with all of these publications that came out of that, documenting what we've learned about SCN8A that then guides treatment. And I think the next phase as well is that this data is critical for drug developers. Drug developers want to better understand the kind of burden there is for seizures, among other things. But seizures, as Michael presented very clearly, we don't know enough about them to be able to truly report, what the seizures look like in SCN8A. And this potentially is information that we can use, that I can use when I talk with drug companies to advocate and say, this is the perfect community for you to focus your energy on for the next, in your drug development. This is a community that has a real burden. There's a lot of need. I mean, those numbers that 70% of our community is drug refractory, medication refractory, that says a ton. And then if we can add to that by saying, this is what seizures look like, this is the kind of seizures, this is how long they last, this is how often they need rescue medication, these are the drugs that work, these are the drugs that don't work. The more power we have behind advocating for increased number of drugs for SCN8A, and then potentially new treatments on the horizon. So I think it's a very powerful tool.
Yeah, and the thing about this is that we applied for an FDA grant a year and a half ago, and we missed getting it by a hair, and that was going to be a 4-year prospective study. And then we thought, well, we'll do a 1-year pilot using Helpilepsy, and then go back to the FDA to try and get the 4-year, $1.5 million grant that'll make us be able to allow us to do this at a larger scale. But meanwhile, in the roadmap meeting, Working Group 3 went off with just the question of how do we improve current therapeutics while we wait for smart drugs. And they came up on their own with this idea that, gee, we should just ask people if they're having a seizure every day and how much that would help. And so just not coincidentally, but it's just a convergence of ideas at this moment in time where this is what we need to do. And it's not being done broadly across many communities. So we're kind of on the cutting edge, which makes it very exciting. And because we already worked on the citizen data, we've written code. We're very comfortable writing code to make nice infographics that display longitudinal profiles of each individual. And it's the individual story in the end that matters. I mean, we can get cohort data like from the registry, but it's usually averaged across the population. We can try divide people into subgroups whether you're mild, moderate, severe, or whether you're a DE, an EE, or a DEE, as we most recently did. But it's still, the individual is going to be different than the neighbor, even within those subgroups. This is our way to get personalized medicine. And it's the first step. It's not going to yield personalized treatment right away, but it's the way to get there. Hopefully some people decided they want to try it, and we'll get an email from you saying you've downloaded it, and we will then onboard you into our weekly data dump and get your data and start incorporating it. Any other discussion?
There are some other questions. Somebody just joined. It says they're Tammy, but they're not Tammy. Has there been any commentary in this meeting on Reaver at all? Mouse model proof of concept-based editing, or Hoffman et al. Off-label anti-inflammatory drug impact on chronic epilepsy? I'm not sure who that is, but Tammy too.
The two studies that were published recently, is that what they're about asking about?
Yeah, I think they're just asking as a research update meeting, I guess, about whether there was any commentary on
Well, the first one sounds like CRISPR editing. That's a long way from making it into human populations. That's probably done in the mouse models. In my opinion, it's still not a perfect strategy because there's something called off-label targets that CRISPR sometimes hits. So it's not perfectly selective. Elyse, you might know something because you were in the industry for a while.
Yeah, there's a lot of off-target effects for CRISPR editing. So my background is I was an industry scientist in the gene therapy space for 10 years. I worked there's a company called Intellia. They've had successful edits in a different disorder, but one of the things about SCN8A is that, and successful gene therapy is that it needs to be localized to a specific area. So most of the areas that have had success are like the eye for macular degeneration, or a different company that I worked for, Voyager Therapeutics. We had some success for even a single base editor. There are some potential. Okay, use inosine still. It's still a long way, especially for SCN8A, because we have a wide spectrum of variants. So as you all know, when you get your genetic report, you have a P and a gene number, and those vary throughout the whole gene of SCN8A. And because there are so many variations within this population, you're not talking about it as therapy. I guess I'm curious to what you're asking, but I'll continue talking about this because there are so many variations that you can have within the SCN8A population. There is no one specific way to target. Well, you can change the whole gene, but because SCN8A NaV 1.6, right, the sodium channel is expressed throughout the entire body, it's very difficult to target.
Anyway, maybe this person can, Tammy
Yeah,
number two can.
if you want to come on and speak, we're just a little curious what you're asking.
Yeah, this is Tyler Youngquist calling in. I don't know
Hi,
how
Tyler.
it has me as Tammy on here. I just clicked on the link for the meeting. The reason is I realize it's completely far away. The inosine base editor is much lower indel rate than the other one, but it's more so what the stage that they tested it on in the mice. So that was my question around the commentary there, because it was.
Are you just asking like the severity of the disorder in the mice?
The impact on the disorder in the mice, because they targeted it before seizures, but it's like a remaining outstanding question of after the seizures, because that is also would also be a good question around treatments, because or if something like that, or something that could help with 8A after the seizures start, what help would be for the condition afterwards, because they did see dramatic improvement compared to the non-treated mice, but It's a question of, well, where did it stop the damage at the beginning, right?
Right. Yeah,
So,
Tyler. So I just did a fairly deep dive on reviewing mouse models for transgenic mouse models for ion channelopathies. I just submitted it to a journal, and it's both sodium and potassium. There are 47 different models that I looked at, and for those models where they were able to do a gene therapy, whether it's an ASO, an AAV, or something like this, there's no study that's been able to treat mice after seizures start and get full resolution without having to go back and repeat the treatment regularly. If you treat before seizures start, that's a different story.
Yep.
So I think that's the state of the art today. Whether that's changed, we don't know.
Also, depending on the phenotype, there's a wide window of when seizures start, and we really don't know. We can't tell, a child is born. Sometimes they're having seizures right away. If they're not.
And with the mice, it's all just limit in the mouse model specifically. It's all just limited as to the limited number that there are. I think there are five specific variants that have mouse models. You've got 1768, and then you've got 1872, and then you've got 1620, and then you've got one of the null variants, and then I think there's one other one.
There's some new ones. There's 850 that's coming along. 1617's coming along.
Okay.
767's coming along. So yeah, there's some new ones coming on. Some of them show behavioral effects too, so that'll be nice to be able to treat behavior and movement disorders. So that there's some advances there as well.
Yep. And I was going to say my second question around the other article that was more off target. I know there have been talks around like angiotensin receptor, but this one was on a medication that just anti-inflammatory, trying to impact chronic epilepsy. Now it's not specific to 8A, but
what's the medication? For the treatment? Is it an anti?
Xeljanz, rheumatoid arthritis.
Oh, okay. Yeah. So there are people working on targeting neuroinflammation with like antibodies or specific anti-inflammatories, that block the sites that block certain sites in the neurons that might be affected by inflammatory agents. But that's promising, but I don't know of anything that's working really well. In the case of our study on candesartan, I'm very interested in promoting that as something people try, and I think we're going to have to do a trial of some sort. But it works in two ways. It works as an anti-inflammatory, and it's also it promotes what's called PPAR activity, which is metabolic. So we know that a huge aspect of epilepsy is metabolic, mitochondrially related and metabolism of glucose and lipids and things like that. And that's probably why the ketogenic diet works in some people. So it promotes that, and it blocks inflammation, and it's indicated for hypertension, but you use it at such a low dose, it doesn't affect blood pressure.
I guess my question with that is, and I hope I hope that you guys are able to get it for some sort of trial or get enough people to buy into it to at least test it out because it has low side effects. Is there, considering the biobank study around, I think it was like the incidence of epilepsy among those adults with high blood pressure, that kind of indicated the angiotensin receptor blocker group as being something that was correlated with lower incidence of epilepsy. And at that time, they tended to focus on losartan, or I don't know how to pronounce that. Do you think there's any way to kind of parallel it with that, saying, hey, there's already some evidence that maybe there's a potential?
Yeah, I mean, that to me, so like I've been working on this mouse model in candesartan for years. And then just near the end of the study, when I was publishing it, two epidemiological studies came out. Millions of people have hypertension and take antihypertensives, and a subset of those also have epilepsy. And so they looked at which drugs they were taking. There's like three major different classes of drugs you take for hypertension. One of them was the angiotensin receptor blockers, which is what candesartan is. And those patients did better. It was losartan, but not candesartan, but it's a very similar drug. And those patients got better with their epilepsy as well. So to me, that's just like, oh, that's really good evidence. It's going to translate into humans, from the mouse to the human.
Yep.
Those were adults, and ours are kids. And so there's probably not a lot of kids taking antihypertensive. So we probably can't do an epidemiological study, but we can do a study with candesartan because it's approved, FDA approved to 1 year old. It's very, very safe. But I know people are reticent to try things, and you have to ask your doctor to prescribe it. If your child has headaches and you know it, it's also for headaches. So you could get a double benefit. But I'm going to have to figure out how we can get doctors to prescribe it. And then I would love just to have people who are using Helpilepsy add candesartan in at some point, and they'll have a before and after to see how it really helped. That's my idea for a clinical trial, to do it with a dozen families or so who are willing to try it and have their doctors prescribe it. It's safe, and you could start out at a super low dose. And I take it myself. I take 8 milligrams a day for my migraines, and it really helps. We could start kids out on 1 milligram a day, and it's already FDA approved. Like I said, it's so safe.
So I just want to also capture a question that came on early in the meeting that we deferred. There was a question about lacosamide as I'm trying to scroll back to it to find it. Oh yeah, that it's a newer sodium channel blocker that only went generic about 3 years ago. And it seems like there may not be a lot of patients on it currently, but do we think that it might be a preferred starting drug in the future that might come up against carbamazepine and oxcarbazepine?
That's an interesting question. We just submitted a paper at Epilepsy that's under review right now, and we used a model to try and predict the best drug based on what you guys said in the registry. And lacosamide came up as one of the top drugs to complement the other sodium channel blockers like oxcarb, because lacosamide works on slow inactivation, whereas the others work on fast inactivation. So they complement each other. And it turns out that lacosamide is an excellent add-on drug. Whether it actually may help some as a first-line drug, but what we're trying to understand is who would benefit as first line and who would benefit as an add-on. And that's what the paper's about. So I think it's a very promising drug.
So my son did attempt lacosamide, but he's also on carbamazepine and lamotrigine, which are two medicines that it interacts with. And unfortunately, trying to ask the neurologist if there's any way we could just replace it with just lacosamide. Like, it's been hard to try to get them to work with me as to what medicines to try or not try. But I really believe that if lacosamide would be tried by itself, it might work.
Yeah, it's possible. I think, again, we're back in the trial and error stage, so it's hard to predict which patient will respond to which drug. But statistically, we're seeing that lacosamide is really helping some people, like I said, either as a first line or as an adjunctive medication. And it's new, and it's not widely used, but more and more people are using it. And so I think it does have a lot of promise.
And there was another question about which one we heard a lot about, whether seizures are the reason for developmental milestone delays or regressions. Is it the seizures, or can you still have delay even if seizures are well controlled? Is it a function of the dysfunction?
Yeah, so we published a paper last year where we used the registry data and machine learning and identified 3 different statistically distinct groups within the population of gain-of-function patients. And some of them had what we call a DE, a developmental encephalopathy. Some have an epileptic encephalopathy, and some have a combination of developmental and epileptic encephalopathy, or a DEE. The community in pediatric epilepsy had determined that DEE was the most appropriate catch-all for kids with these disorders, but we really feel that some kids do have an underlying pathology that stymies developmental progress, and seizures are part of that process, but not the major part of it. Whereas some kids have epilepsy, seizures that secondarily cause developmental delays. And so if you're in the DE group, it's easier to treat seizures, but you'll still have some problems with development. Whereas if you're in the EE group, then knocking the seizures down really helps accelerate development. In the DEE group, so then helping seizures has milder effects on development, but some. So it's always good to get rid of seizures if you can, whichever group you're in. But development seems to be the developmental encephalopathy seems to be its own thing, underlying some of the other symptomology of the disorder. So you're right on with that question.
Hopefully you heard those answers, Peg. I know you wrote you had to drop away, but if not, this is recorded and you can watch and catch up on everything. Can you repeat, Michael, the drug for the headaches that you were talking about?
Yeah, it's candesartan, C-A-N-D-E, sartan, S-A-R-T-A-N.
Maybe Elyse can put the paper you wrote on it in the chat. That would be helpful, I think.
Yeah, absolutely. We published it just recently.
Yeah.
I can try and find it too.
So, I mean, there's a good follow-up question that Peg asked. It's like, how do you tell whether you're DEEE or DEE?
Yeah. So the simplest rule of thumb is you ask yourself, when did developmental delays start and when did seizures start? It's not the only way of figuring it out, but if developmental delays started a few months before seizures, or if seizures started months before developmental delays were noticed, that might put you into one or the other category. And we also know that loss of function with or without seizures is a developmental encephalopathy because some kids don't even have seizures, but they have developmental delays. And so we know, SCN8A, whether gain of function or loss of function, is very much a developmental disorder with seizures as either a secondary outcome or as complicating the developmental profile.
And I would suggest, Peg, I know you're new to the whole community. I'm going to suggest you also send an email to Michael, and you can share your genetic test results and share what your concerns are. And we might be able to tell from your specific mutation, whether it's gain of function or loss of function or DEEE, those kinds of things. Some have been classified, some hasn't. So I just put the email in the chat.
Also, I put the paper in the chat, the PDF, so you can read the paper on candesartan. Please let me know if you're interested in that and seeing if you're interested in using it, because I do really want to get a little trial together, and not like a big clinical trial like the pharmaceutical companies do, but something on a smaller scale where we look at a period of time before you take it and a period of time after. And my prediction is if it works, it will make the time between seizure bouts longer, and you'll have improved non-seizure outcomes in the interim. It's not an anti-seizure medication. It's something that protects the blood-brain barrier from seizure activity. So a lot of the downstream effects of having seizures are that your blood-brain barrier is disrupted, and then once that happens, you get more inflammatory and a vicious cycle of creating more seizures. And this drug sort of cuts that off at the pass.
What qualifications do you need to participate?
Basically, I think it can help loss of function too, but I think anyone who wants to try it, you might have to talk to your doctor. Your doctor might say, I'm not going to prescribe something that's for hypertension, but show them this paper and then see if that changes their mind. But anyone who wants to try it, I think can try it, because if it's protecting the blood-brain barrier, I believe both loss of function and gain of function have effects on the blood-brain barrier, disrupting it, as do many other neurological disorders. The blood-brain barrier is a common feature, as is neuroinflammation among many neurological disorders.
So you said neurologist or a primary care physician?
Whoever's prescribing your meds, probably best to talk to neurologists, but.
Okay. What age group?
Well, it's safe to 1-year-old. So yeah, anybody can take it. Like I do. I take it. It has no side effects that I notice. We haven't tested it. So your doctor might say, well, it's never been tested in young kids with seizures. Well, that's true. So that might be that's why I think like starting at a super, super low dose. And if it is FDA approved to 1 year old, that means it has been tested in children.
So I don't think there's, if I missed something in the chat, please let me know. Otherwise, I think we can hear from other families if they want to come off mute and ask a question, or we're here. So I also just want to add, if there's anybody who's not yet part of our private Facebook group, I'm going to put the link in the chat to join, because we would love to have you there. We do a lot of support and ask a lot of questions and answer a lot of things on there. Because I see some new names in here, and we're so glad that you're with us. Anyone else have a question or want to give us an update on how they're doing?
I think my last Hammer Lab update was on candesartan, so that might have been recorded. Was that recorded, Gabi?
I think so. I can double check.
Yeah, because that be and I'm happy to give it again. I talk about some of the things that Tyler mentioned with the epidemiological studies as well as the mouse study, and what the mechanism is and how it's protecting the brain from seizures.
Yeah, I think maybe we'll do another research update in a couple months. And, I think there will be some emerging information coming out about some other breakthroughs that are happening. There's a paper that's just uploaded on bioRxiv, so not yet reviewed and published, but about base cell edit. What is it called? Cell base editing. What is it called,
Base
Michael?
editing, yeah.
Base editing, yeah. Which is a new kind of breakthrough, and
That might be what Tyler was talking about.
Well, I didn't recognize those authors, maybe.
Okay.
So
'Cause
how,
I thought that was,
so how
what?
convince a neurologist like Liam's neurologist to prescribe something like that when like I asked him about the meds, and he just said because it wasn't FDA approved for epilepsy. So how do you convince them that it could possibly help the inflammation in the brain and help the seizures? Because I believe it probably could help, but it's hard to get them to, with pediatrics, it's hard to get them to do stuff like that. Is there a way to have a study or something?
Yeah, that's what I'm trying to learn. I'm kind of a novice when it comes to trials, but maybe there's some way we could. I don't know. It is going to come down to the clinicians prescribing it, because we're not going to get, it's an off patent, it's generic drug. So no company is going to say, oh, I want to invest in this because it's a moneymaker. It's not. So we're going to have to do it as a generic off-label drug. And that comes down to doctors prescribing it. It's cheap, it's available, and it's going to have to come down to like a dozen families trying it and convincing their doctor to give it to them.
Do you happen to know any interactions with seizure meds offhand?
It doesn't have any known interactions. I'd say take the paper and talk to your doctor about it. And I guess we could also find the papers that we were talking about, the epidemiology, where it showed that in humans, it improved seizure outcomes in patients that took it for hypertension. I can try and find those articles as well, make them available.
Even if there is no hypertension available, in the history?
Yeah. Well, that's the thing. The risk is it's been tried in kids for hypertension, but it's never been tried in kids who have epilepsy for safety. But there's no reason to believe that it would be not safe for kids who have epilepsy. But again, that's why we'd have to be careful and start out at an extremely low dose and titrate it up to something that should be therapeutic for protecting the the blood-brain barrier.
Dolan could take, even though he doesn't have seizures, he has that abnormal brainwave activity that increases at night. And then, like, he does have migraines. Is that something they could almost watch on an EEG if he takes that before bed and compare the like abnormal brainwave activity, like with it in
What are
his
his
system
other,
versus off?
what are his
I don't know, I'm
behavioral,
just thinking.
yeah, no, what are his behavioral symptoms other than abnormal EEG? Like what do you notice about him?
So he has loss of function, and so he has he's low tone, he has apraxia of speech, he has he's diagnosed with autism, he has the vision therapy assessment where he was below the 20th percentile in 9 out of the 10 areas. So he has a lot of that type of stuff going on.
How often does he
And
get
then,
headaches?
yeah, just like
How
daily.
often? Daily headaches?
Daily. Yeah,
Oh,
daily.
wow. Yeah, then I would say, and does he take anything for that?
No.
Okay, that's a great thing to try for headaches. It's level 2 for headaches, for migraines.
Okay.
Candesartan is, so you can ask your doctor for candesartan for headaches. And if it helps his headaches, that's a win right off
Okay.
the bat. Then you can see if there are any other changes that go along with taking candesartan, like improvement in behavior, alertness, other things going on. Yeah.
Okay. Thank
Yeah.
you.
Sometimes we don't know if our kids have headaches if they're nonverbal, but we do hear from families quite often that headaches are an issue.
Yeah, he has them so bad that he'll get sick to his stomach.
Oh, that's very migraine
Yeah.
sounding. Yeah. Yeah, give it a try. I used to have bad migraines and now I don't. Migraines are not unlike seizures in that it is an electrical activity in your brain. It's just more superficial on the brain rather than deeper in the brain. But it's a paroxysmal spreading event, they call it, and it's similar to what a seizure is. What's the name of the migraine? Oh, it's candesartan. Edan, I put the paper in the chat. It's a PDF if you can grab it.
Could that possibly be the quote unquote abnormal brainwave activity that they're seeing increased at night because it's the end of the day and his body is just trying to get rid of that migraine?
Yeah, I don't know. That's a thought. I don't know exactly what his abnormal brain activity is and what causes it. And I'm not sure people understand very well what causes abnormal brain activity in an EEG setting. Some doctors treat the EEG, which is interesting. Like an old-fashioned sort of treatment is like, let's look at the EEG and then look at a medication if it changes the EEG. I don't know if that's as effective a strategy as treating, let's say, seizures or something else, but some people will use the EEG as a way of assessing whether medications are working. I guess they expect us to go back to baseline physiology, normal physiology, and I don't know if that's common or not. One of the things I'd like to do in the citizen data is to see of all the EEGs that people take over their, the course of months and years, how often does an EEG change from normal to abnormal and abnormal to normal? Like, EEGs are not perfect. They can miss abnormalities because they are superficial. But does the EEG ever change for the better with treatment? And I don't know the answer to that. Maybe some of you have experience where the EEG has gotten better.
The EEG got better the second night we were observed because he took Zofran because he was getting sick to his stomach again from the migraine.
Well,
So there was
good.
less abnormal activity.
Well, that's interesting. I guess you'd have to do it multiple times to make sure that that's the cause. Like correlation is not cause, but doesn't prove causation, as they say. So we'd have to kind of look at it multiple times in multiple individuals to see if, in fact, helping his migraines with a drug actually changes his EEG in a positive way.
Any other questions or ideas? Well, this has been a amazing discussion. Thank you all so much for joining. Do you want to clarify the session on Tuesday, what and when that is? That was in the chat.
The session on Tuesday is just intended for those, we have a couple data scientists and some of the team at U of A to discuss alternative data collection sources and how we can harmonize that with our current Helpilepsy data.
Ah, okay. I didn't
So if
realize
you're
it wasn't
already-
in the chat, but it's basically for data scientists, people, not for anyone who might be-
Anyone collecting their own data that wants to learn how to share it with us.
Ah, got it. Okay. And is the link and the time in here if anybody wants that?
No, they just need to email me and then I'll share it with them.
Okay. All right. Well, thanks everybody. We're right on time. Appreciate y'all coming out. I think I counted that we have 1, 2, 3, at least 5, 6 different countries on the line today. So lots of global excitement about this. So thank you all for joining, and the email and the chat will get you in touch with Michael if you have questions. And yeah, we hope to see you again soon. Stay in touch, email us, and stay well. We'll talk to y'all soon. Oh, and for anybody who doesn't know it, we currently have an SCN8A art auction going on. I'm super excited about this. It's art from a lot of different SCN8A families and people living with SCN8A. So please check it out, share widely, help us spread the word. The funds that we raise here are going to be put towards research that is coming out of the research roadmap, the priorities that families put forward in preparation for that meeting. So thank you all for contributing, for bidding, for sharing, and we will see you all next time. Any other announcements? No? Okay. Thanks, everybody.